This final video is all about anaerobic respiration and fermentation. So quick recap here. So we've talked about aerobic respiration. Okay, so let's go ahead and write down anaerobic because that's what we're going to talk about here momentarily. And then fermentation. Okay, so remember with aerobic respiration, there was glycolysis. And then we had that linking step, which is called pyruvate oxidation. And then we had step 2, which was the Krebs cycle. And then we had oxidative phosphorylation. Okay, so I'm gonna abbreviate that OPP. And remember, y'all, that had the electron transport chain and then the chemiosmosis part. Okay, and so when it comes to anaerobic respiration, it also has glycolysis and pyruvate oxidation. I'm going to kind of abbreviate these a little bit. And then it does have the Krebs cycle, and it's got oxidative phosphorylation with the electron transport chain and chemiosmosis. Okay, and what you're going to learn about momentarily with fermentation, it's got glycolysis. Okay, and then it's going to have something else as well, but we'll get to that momentarily. So, okay, so if you look at aerobic respiration and anaerobic respiration, you see that the overall name of the steps and the gist of the steps are the same, right? People tend to think that aerobic and anaerobic respiration are wildly different processes, but they're not, because they're both respiration, right? The big difference is the aerobic versus the anaerobic. And remember, aerobic means oxygen is present. It requires oxygen. And that was because down here at step 3, oxygen was the final electron acceptor. Okay, so anaerobic respiration is going to occur when oxygen is not present. So therefore, oxygen cannot serve as the final electron acceptor. So that's where the big difference comes in between aerobic and anaerobic respiration. The final electron acceptor is normally going to be either sulfate or nitrate. Okay, so those are other molecules. So if you remember from chemistry, oxygen is, what's it called? Is it electronegative? Where it's kind of greedy and it likes to accept electrons, right? Well, sulfur and nitrate are very similar in that sense. They're happy to accept electrons. So at the end of the electron transport chain, anaerobic respiration is going to use either sulfate or nitrate. Now, I do want to point out, though, that right, organisms that use anaerobic respiration are going to be pretty different than organisms that do need oxygen, right? Because their environment is different. One has oxygen, one does not. And so even though they have these same steps, which have the same purpose, the exact proteins and enzymes and things that are involved in each of these steps is going to be a little different, because different enzymes are needed in different environments. And so like the Krebs cycle in anaerobic respiration is much shorter and more condensed than aerobic respiration. Therefore, there's about 50% less energy, 50% less reducing power, and therefore less energy produced during anaerobic respiration. Okay, just because it's a slightly different version of the same cycle. Okay, so as you can see, though, with fermentation, the only thing all three processes have in common are glycolysis. Okay, And so if you are following along in the slides, I'm going to go up to this one real quick. You'll notice that there are two different types of fermentation shown on here. But what do all fermentation reactions have in common? Glycolysis. Okay. And so I'm going to use a different slide here. So give me 1 second to add in a new slide. Okay. So for fermentation, I'm very scientific when I talk about it. So this is typically an anaerobic process. Anaerobic. Okay, well, there's no oxygen or very little oxygen present. And so when I think of fermentation, I usually think about glycolysis plus some other reaction. That's how I describe fermentation. And here's why. So with glycolysis, okay, so remember, that's going to be glucose, and it's going to get broken down into 2 molecules of pyruvate, 2 molecules of ATP. That should be a plus sign. I'm sorry, y'all. I don't know why I put a comma. And 2 molecules of NADH. Okay, so that's the glycolysis part. And so this is the only step during fermentation that is going to produce ATP, okay? But we actually have a slight problem with this situation. And that is going to be that we've made 2 molecules of NADH. Now, when we were talking about respiration, NADH was really important, right? It was that stored energy because it dropped those electrons off at the electron transport chain. But look at this previous slide. Is there an electron transport chain involved in fermentation? No. So now NADH is actually problematic because we have to drop off the electrons that NADH is holding on to. And to better see why, I need to expand out this glycolysis reaction, okay? So remember, we do start with glucose. I'm going to abbreviate to get myself a little bit more space. So GLU is glucose. But remember, in order to make ATP as a product, we needed to start with ADP. And I'm not worried about numbers right now. I'm just trying to help you understand why the NADH is problematic. And so in order to make NADH, what does that start out as? NAD plus, right? And so in order to make the pyruvate and the ATP and the NADH, we need these other starting molecules, the glucose, the ADP, and the NAD plus. And so in order to generate that ATP, which these organisms need to survive, we need a surplus of ADP and NAD plus, right? Because they're required for those energy molecules. Well, because there's no electron transport chain, this NADH as a product, it's just going to sit there in the cell because there's no electron transport chain to drop it off at. Okay, so eventually glycolysis is going to come to a standstill, because there's going to be no more NAD plus because it's all tied up in the NADH, okay? So these other reactions that I mentioned up above, their main purpose is to use that NADH, okay? In other words, they need to oxidize the NADH because they need to strip it of those 2 electrons. Because once it's stripped back down to NAD plus, it can then be used for glycolysis to keep producing ATP. Okay, so the other reactions are also going to form some sort of important product that the bacteria or whatever needs. But in order for that organism to stay alive, it needs to oxidize this NADH. Okay, so when you look at the slide in the PowerPoint, okay, so you'll see the glycolysis up here is getting turned into pyruvate. People also call pyruvate pyruvic acid, so it's the same thing. And then notice that over here in lactic acid fermentation, do you see how NADH is going into the reaction to make NAD+? Well, this NAD+ can now go back up and be used in glycolysis to generate that ATP. Because remember, organisms that depend on fermentation get 2 ATPs total from each round of glycolysis. Okay, respiration was making up to 32 ATPs. Fermentation only gets 2. It is very minimal ATP production. So they are dependent on glycolysis functioning properly. If you look down here at alcohol fermentation, all right, you'll notice the same thing. NADH is going into this reaction in order to regenerate the NAD+. That again goes back up here to glycolysis, okay? And so each of these has to oxidize NADH, and then they are producing things that the cell needs, like lactic acid or ethanol. Okay, I'm not going to ask you, what's made during alcoholic fermentation, after pyruvate. I'm not going to ask you to remember acetaldehyde or whatever, but you do need to understand the importance of fermentation. And then I do suggest you fill out this chart as a way to review these processes. This is something we will go over when we do the exam review. All right, y'all, that is the end of the bacterial metabolism information. Let me know if y'all have any questions.